School Quiz students. I'm a biology teacher Swathi, and in this video lecture, we are going to study the unit, the fundamental unit of life. This chapter has been included in class. Up till this portion, we have seen the basic definitions of cell theory, types of organisms, and what all the types of cells are there. Like the cells can be classified on basis of their shape, sizes, prokaryotic cell, eukaryotic cell. Then we have seen the difference between plant cell and animals. After that, we saw the various type of movements which are there in cell, like diffusion can be there, osmosis can be there. Then we saw the type of solution that are hypotonic, hypertonic, or isotonic solutions. And now, before proceeding to the further part, we should just discuss the exercises and the portions which have been included in your syllabus so that we can just get to know that how much we are actually able to grasp with this. Okay, so and like students, this is the best way to assess also that what all have you understood till now. Okay, and I hope that like you are enjoying these lectures too. So the first question is, who discovered cell and how? So as I told you before also that cell was discovered in 1916 by Robert, and he discovered it by while examining a thin slice of core through self-designed microscope. So he himself only designed a microscope by help of which he was able to examine the thin slice of core, and he observed that inside the core there were some hexagonal structures like a honeycomb, which were consisting of tiny compartments. So those compartments can be compared to a room also. So like honeycomb shaped rooms, for various rooms were there. So that was, these minuscule boxes are referred to as cells. So these cells were actually in the shape of small compartments only. Okay, and so the name of scientist is very important. So it was discovered by Robert Hooke and in the year 1660. Next is, why is the cell called the structural and functional unit of life? So since starting of this unit, we have been repeatedly saying that cell is structural and functional unit of life. So cell basically forms the structure of an entity. Like suppose if something is spherical shape, or suppose we say that this mobile is rectangular in shape. So there is something which is providing the structure to this. So in any living organism or in any entity, cell is there to form the structure. So a group of cells will combine to form a tissue, and tissues will combine to form organ, and organs will combine to form an organ system. And then further, they will perform fundamental functions like various life processes can be there, like they can be involved in the process of respiration, in the process of digestion, excretion, and these both processes can be seen in both the cases, like whether unicellular organisms are there or multicellular organisms are there. And all these activities are performed independently. That means that these cells are not dependent on each other, so that these should be performed. In short, we can say that cell is something which is providing a fixed structure to a living organism, and various type of functions are performed independently inside the cell. Hence, we can say that cell is the structural and functional unit of life. Next is that we have seen that plants take in carbon dioxide and they release oxygen, and that's because of which we say that we should plant more and more trees, so that if we will plant more and more trees, they will release more and more oxygen, so we will be able to have more free oxygen in the environment. But how actually this happens? The movement of CO2 and water take place in and out of this. So very mainly the movement of gases take place by the process of diffusion. So these cell waste accumulate in the high concentration in the cell, and whereas the concentration of CO2 in the external surrounding is comparatively lower. So we know that plants take in carbon dioxide. So it is obvious that the concentration of carbon dioxide inside the cell will be higher and outside the cell will be lower. So this difference in the concentration will create an gradient. That means at one position we are having high concentration and on another point we are having low concentration. So a gradient or a difference is created. And this difference in the concentration level inside and outside of the cell will cause the CO2 to diffuse from a region of higher concentration that is within the cell to the concentration which is low that is outside the cell. So because of that difference in the concentration, only the movement of carbon dioxide has been seen. Okay, and now if we talk about water, so water diffuses by the process of osmosis, and the major difference between diffusion and osmosis is that in osmosis the exchange basically takes place with the help of a semi-permeable membrane. So water diffuses by osmosis through the cell membrane or some membrane should be there. It moves from a region of higher concentration to a region of lower concentration through a selectively permeable membrane until the equilibrium is reached. So it will move from inside of the cell to outside of the cell. Now you will ask that, ma'am, why does it happen that like any of the substance always moves from its high concentration to low concentration? So student, this happens because it is a normal tendency of anything. As you know, suppose if you are standing on your terrace and you are just throwing a bucket full of water downside. So it is very easy for you to throw the water on the ground. But suppose if you are standing on the ground and you have to throw a bucket full of water to the terrace, is it feasible for you? No, that is not feasible for us. So because like it is, if we have to take something from lower level to higher level, there needs to be some expenditure of energy or some external sources required so that that energy can be provided and that substance can move from its low concentration to higher concentration. And as we know that diffusion and osmosis both are the passive process in which there is no requirement for any external energy or ATP. So because of that, normally the processes, this you should make a thumb rule that if there is any process in which energy is not involved, so it is obvious that at that any substance is moving from its high concentration to low concentration. That is only possible. Okay, so in that way the in and out movement of water and CO2 have been seen in the cells. Next is we have to fill the difference between this prokaryotic cell and eukaryotic cell. So here you can see the size is generally small for the prokaryotic cell, that is 1 to 10 micrometer, and 1 micrometer is nearly equal to the 10 to power minus 6 meter. If we talk about eukaryotic cell, so the size is 5 to 10 micrometer. Okay, so it is from 1 to 10, and like for eukaryotic the range starts from 5 to 10. If we talk about nuclear region, so the nuclear region in case of prokaryotes is poorly defined because there is absence of any nuclear membrane and known as nucleus. Whereas if we talk about eukaryotic cells, so the nuclear region is well defined and it is surrounded by the membrane which is known as nucleus. Okay. If we talk about chromosome, so in case of prokaryotes, we have single chromosome because in prokaryotes mostly asexual reproduction only takes place. So because of that, only genes from one parent has been inherited by the offspring. Whereas if we talk about eukaryotic organisms, so there are more than one chromosome present in the case of. If we talk about membrane-bound organelles, so membrane-bound organelles are basically absent in the case of prokaryotes, whereas membrane-bound organelles are present in the case of. Now, as we have seen that what are the differences which can be seen between different cell types. So like there can be a major difference between cell division also if we talk about. So what type of cell division is required for growth and repair of bone? So we know that if any growth or repair is going on in the cell, so it is obvious that the cells are dividing repeatedly so that like previous cells have been like replaced by the new cells. Okay, and which is the division in which the formation of gametes also takes place. Okay, so there are two ways by which a cell can divide. One is mitosis division, in which the equational division of the cell takes place, and another is the meiosis, which is known as reductional division. So meiosis is the type of cell division that is involved in the growth and repair of the cell. Okay, because it is an equational division. So if we are having 2n organisms, so after division we will get 2n and 2n organisms. Okay, but if we talk about meiosis, so meiosis basically is a reductional division. So if we are having 2n cell, so after division we will get n and n. And that is what is required in the formation of gametes, because like for male have X and Y chromosomes, and if we talk about female, we have X. So if the fusion of gametes have to take place, so there is probability that the X from male can combine with one of the X of the female, or Y of the male can combine with one of the X of the female. So in that way, if we take complete as a 2n, so n for X, n for Y, n for X, n for like n can be there. So if they will combine, so in combined way they will form 2n. So because of that, to maintain that fluidity, the process of meiosis is required, so that the number of gametes can be reduced to the half. So whereas meiosis is the type of cell division which results in the formation of gametes. So this is very important that you should remember the significance of mitosis and meiosis. So the mitosis basically is required for the growth and repair of the body, whereas meiosis is involved in the formation of gametes. Next, now we will see an experiment to just check the process of osmosis. So what we will do is, we will take 4 peeled potato halves and scoop each one out to make a potato cup. So what you have to do is, you have to take 4 potatoes, you have to peel them and have to cut them into half, and you just have to scoop and make that hollow from inside. Out of these potato cups, one should be made from a boiled potato. Put each potato in a trough containing water. Then you should take a trough or a tub should be taken, and which should be filled with the water, and in that you should keep that. So then there are certain conditions. First is, keep cup A empty. The cup A should be empty. Nothing should be inside the cup A. Second condition is, put 1 teaspoon sugar in the cup B. Third is, put 1 teaspoon salt in cup C. Fourth, put 1 teaspoon sugar in the boiled potato in the cup A. Okay, so these 4 are the cases. Now we have to keep them as that way for the 2 hours. Then we have to observe the 4 cups and answer the 4. Then we have to explain why water gathers in the hollow wood of portion B and C. So you know that in case of B and C, the concentration inside is higher. So because of that, the movement of water is taking place. Why is the potato A necessary for this experiment? And explain why water does not gather in the hollowed portion of A and B. Okay, so in that way we have to answer this. So if we talk about first, so the water accumulates in the hollowed portion of B and C as a difference in the water concentration is observed. Thereby, endosmosis process acts as a semi-permeable membrane. So inside, the concentration of solute is more, so the concentration of water will be less. So water will move from its high concentration to its low concentration, that is from outside to inside by the process of endosmosis. Second, the potato A is having significance in this experiment to compare different scenarios. Like if we are having some reference, then in reference to that only we can compare other entities. So cups B, C and D. The potato A in this experiment clearly shows that potato cavity on its own cannot bring about water. Okay, so like cup A in this experiment is basically working as a reference for this experiment. And cup in A does not show any change in the water concentration for osmosis to occur, which requires the concentration to be higher than other cells. Whereas if we talk about cells in D, so because of boiling of that cell, the cells in cup D have died, because there is no existence of a semi-permeable membrane for water flow. So consequently, the osmosis will not occur. So from this experiment, we have got to understand that first of the thing is that there should be some concentration difference so that water can move. Another thing is a cell should be alive. Because of that only there will be movement. In that cell, no movement can be seen. And second is there should be existence of semi-permeable membrane. Then only the passage of various substances can be seen. Okay, that's all for today's lecture, students. Keep learning, keep enjoying. Bye bye. Thank you.